Greetings. My name is Julian Torres Rodriguez. I am a senior student from the Pontifical Catholic University of Puerto Rico, and now I'm going to be talking about a project I had the opportunity to work on this summer under the title Methylmercury Alters Gene Expression of AMPA Receptors Subunits on Human-Induced Pluripotent Stem Cells Motor Neurons. The aim of this study was to determine whether acute methylmercury exposure at toxicologically relevant concentrations increased or decreased the gene expression of the AMPA receptor subunits. We hypothesized that methylmercury will alter the gene expression of the AMPA receptors in our human-induced pluripotent stem cells motor neurons. So, as methylmercury concentrations increase, there will be a decrease in the gene expression of the AMPA receptor subunits. First, let's talk about methylmercury neurotoxicity. Methylmercury is a commonly encountered form of environmental mercury due both to its widespread use as well as to biomethylation by aquatic organisms. Mercury sources include factories, volcanoes, power plants, and others. Mercury enters the atmosphere as a vapor, where it becomes part of the water cycle. Once in the atmosphere, mercury can remain in its vapor state or become oxidized and recycled to the earth in rain. Accumulated oxidized mercury is a substrate for methylation by sulfate-reducing bacteria in bodies of water, generating methylmercury and entering in the food chain by which comes to humans. Methylmercury is considered an environmental toxicant that causes sensory and motor dysfunction, and it is known that AMPA receptors are associated with the methylmercury-induced cell death, possibly due to excitotoxicity. Now, what is an AMPA receptor? Alpha-amino 3-hydroxy-5-methyl-4-isosacol propionic acid, better known as AMPA, is an excitatory ionotropic transmembrane receptor for glutamate, the most abundant excitatory neurotransmitter in the brain. It mediates fast synaptic transmission in the central nervous system. AMPA receptors are responsible for primary depolarization in glutamate-mediated neurotransmission and has 4 binding sites for glutamate. They are located in the pre- and postsynaptic neurons and are composed of 4 types of subunits called GluA1 through GluA4, which combine to form a tetramer. GluA2 subunit presence is important for calcium permeability of the receptor. It has been reported that methylmercury affects motor neurons by increasing the intracellular calcium concentration. In addition, it is known that the intracellular calcium increases occurs through AMPA receptors, so it is believed that it leads to excitotoxicity by the overexcitation of the glutamate receptors. To achieve our aims, at the in vivo number 7, we expose ourselves to different concentrations of methylmercury. The human-induced pluripotent stem cells motor neurons are a human cell line that were obtained pre-differentiated originally from the peripheral blood of an adult male that expresses typical markers of motor neurons. After methylmercury exposure, we perform the RNA isolation, nanodrop of the extracted cells, RT-PCR, nanodrop again, and quantitative PCR using TACMAN as a probe. Here I am representing the gene expression that was measured immediately 1 hour after methylmercury exposure. We exposed the human-induced pluripotent stem cell motor neurons for 1 hour, and we measured the gene expression immediately after the methylmercury exposure. In the y-axis, you will see the fold change compared to the control, so the untreated cells, and the housekeeping gene, which was GAPDH, glyceraldehyde triphosphated dehydrogenase, a well-known housekeeping gene which is used as a biological control because of its stability after any condition. In the x-axis, you will see the methylmercury treatment, and the exposures are in increasing concentrations. So basically, what we see here is that at 0.1 micromolar of methylmercury, the cell increased the overexpression of the subunit studied. These do not occur significantly until we get to the highest concentrations. So maybe the cells are trying to compensate if methylmercury is possibly interacting with the receptor by increasing its expression. However, we expose the cells for 1 hour and then wash out methylmercury. Then the gene expression was measured 24 hours after the beginning of the exposure. And what we observed is that actually there is a downregulation of the genes in all the concentrations except at 0.1 micromolars. Maybe this is due to the increased cell death that we observed after methylmercury exposure. So after all this, in summary, we can say that immediate expression after exposure shows that there is an increase of expression of all the subunits at 0.1 micromolar to 1.5 micromolar mercury exposure. And after 24 hours, the effect of methylmercury in gene expression seems to be irreversible because of the decrease observed in all the concentrations, except at the lowest concentrations, which is at 0.1 micromolars. In summary, our results demonstrate that methylmercury causes a differential effect in the expression of the AMPA receptor subunits in a human cell model. These preliminary results are relevant because it is the first study that demonstrates that methylmercury affects, by a concentration-dependent manner, a human cell model. To conclude, I want to say that I am eternally grateful to my wonderful mentors, Alexandra Colon and Kimberly Rivera, for all their commitment and excellent teachings. It has been a very enriching experience. I would like to thank Dr. William Atchison for giving me this opportunity, Nicole Colon Carrion, a previous Andrew student, for collecting all the data for this study, and Melissa Jaime Cruz for providing me all the information about the program. It has been a wonderful experience. Thank you so much. So if there is any question, I will be waiting to answer. Thank you so much, everyone.